So what I intend to do today is talk about the melanoma treatment landscape. And I'm going to mostly talk about where we are now and where we need to go in the future. And I will talk about some of the challenges we might have going forward. But I think it's instructive to ask how we got here in the first place. And I can tell you that when I was training as a clinical fellow, I heard many times that melanoma was a disease that couldn't be treated. And that is not the case anymore. So let me kind of illustrate that for you more graphically. Here are my disclosures. So this is a patient I saw 3.5 years ago. And each one, this is a PET scan showing, let me see if I can get the pointer. Each one of these dots is a melanoma, a separate tumor, and you can see her body was filled with melanoma. And this was her scan 12 weeks later. We couldn't find a melanoma, and we looked really hard. And this is what her scan looks like 3.5 years later. She has no melanoma. She's an active 73-year-old woman. And I think it highlights the progress we've made in treating melanoma. Obviously, not everyone responds in this way, but I think it tells us we are making great progress. And this is not just an anecdote. So this past year, it was reported that the rate of cancer deaths overall dropped in the period of 2013 to 2017. And this decrease was for melanoma 7% per year. So we are making progress. We have a lot of ways to go, but I think the path we're on hopefully will cure more patients than the one I just showed you. So I think it's instructive to think about why and where we've been successful in our treatments. So this is a graph. It's a timeline of FDA approvals for melanoma. And by the way, I was really impressed that so many of you correctly identified the number of FDA approvals for melanoma. So what you can very clearly see in the last decade, there's been an explosion in the number of treatments that are approved for melanoma. And this is undoubtedly helping us cure more and more patients. But I also want to make another point with this timeline, and that is this period from 1975 to about 2010, which is often described as a drought. And that's because there were very few treatments that were approved or effective for melanoma. And I would argue that, in fact, during this drought is where the genesis of the current treatments came about. So if we actually look at the preclinical work that led to these treatments, it was during this drought period. And so I think it's important to recognize the engine that will drive innovation in our treatment comes from research. And I don't need to tell this audience about that. But it's a reminder as we think about how do we improve the treatments we have now to even greater success. So I apologize. It looks like some of these diagrams didn't turn out too well, but I'll just take you through the diagrams themselves. So I'm going to be talking about two types of treatment for melanoma. One is called targeted therapies, and the other is called immunotherapies. And targeted therapies work by turning off a switch that drives the growth of cancers. And we know that in about half of melanomas, there's a mutation in this gene called BRAF. And this pathway is present in every cell, but normally in healthy cells, this pathway is kept in check. In contrast, in about half of these melanomas, there's a genetic change in this gene called BRAF that turns on this growth support promoting pathway, leading to uncontrolled cell growth. And we now have targeted therapies, in fact, several targeted therapies that block this protein as well as some related proteins. And these combination therapies have demonstrated efficacy and are now approved for melanoma. Because these treatments, oh, perfect, that's amazing. It's like putting glasses on, you know? It's important to remember that the other half of melanoma patients that don't have this BRAF mutation currently have no approved targeted options, and we'll talk a little bit about that in a minute. So what are the pros and cons of targeted therapy? Well, as I mentioned, we use this therapy if your melanoma has a BRAF mutation. That is to say, we test your melanoma. If it has a BRAF mutation, we can offer this therapy. And for that reason, we see tumor shrinkage in almost all patients treated with this therapy. The effect of this treatment is very rapid. So here's another PET scan. This was a patient with melanoma, and this is the same patient. We did a PET scan 15 days later. And you can see rapid improvement of symptoms. This treatment is largely tolerable, and it's taken as a pill, so it's rather convenient for our patients. There are some downsides to the targeted therapies. One is that it's only available for those patients whose melanoma have the BRAF mutation. So the other half, as I mentioned, will not benefit from the targeted therapies. And unfortunately, we find that in patients who do have the BRAF mutation, who do derive benefit from these drugs, unfortunately, the melanoma finds a way of escaping from these drugs, and that process is called acquired resistance. So an emerging concept we'll talk a little bit about in a few minutes is how do we overcome the acquired resistance that we see in most patients treated with targeted therapies. So the other form of treatment that we use is called immunotherapies. So one can think about this as your immune system is primed to recognize foreign things, be them viruses or cancer cells. And a particular cell type within the immune system is called a T cell. And these cells have the capability of destroying tumor cells. But what happens is that the tumor cell finds a way to turn off the T cell. And it does this by turning on this protein called PD-L1. And when the tumor cell puts on PD-L1 on its surface, it binds to the T cell's PD-1 and turns it off. And so what anti-PD-1 or anti-PD-L1 therapies do is they block this interaction between the PD-L1 and the PD-1, thereby activating the T cell to destroy the tumor cell. So what are the pros and cons of immunotherapy? Well, here's a recent publication comparing 3 different kinds of immunotherapy: ipilimumab, nivolumab, or the combination. And you can see that not everyone benefits from the immunotherapy. But if you do benefit, this curve, sorry. Well, the curve that, what you see is that there's a long plateau, a long tail, and that tail is reflective of the durability of these treatments. So if you do respond to these immunotherapies, many of those responses can be durable. Like targeted therapies, most immunotherapies are tolerable, but these therapies do have some disadvantages, and one of them is that unlike targeted therapies, where we could predict whether you're going to respond to these treatments, we don't have a good way of predicting whether an immunotherapy is going to work for you. And that's because immunotherapy response is influenced by so many factors, including what's happening in the tumor, what's happening to your immune system, and even the bacteria that are in your gut. Immunotherapies are generally given as intravenous, so it makes it a little less convenient, and they also may have a more delayed response compared to targeted therapies. So how do we decide whether to use a targeted therapy or an immunotherapy? Well, I will say that at the present time, we have no direct head-to-head comparison between these types of treatment. And so what I've done here is compared a currently approved immunotherapy to a targeted therapy that's also approved in 2 separate clinical trials. One must be very cautious about doing these comparisons, but I think this comparison does illustrate an important point. And that is, if you look at the benefit of the targeted therapy, you see that pretty soon right after starting the treatment. Whereas the benefit of the immunotherapy in red seems to be delayed. But ultimately, these curves cross over. And that is a reflection of the fact that immunotherapy responses can be more durable than targeted therapies. And so that is often a consideration when we think about tailoring the treatment for each patient. And so the focus of research at the present is not only to shift the curve up, so make, for example, targeted therapies more durable, more effective, but also to shift the curve to longer durability, especially for targeted therapies. So where do we go from here? So I've listed 5 important questions that I feel that we need to address. The first 2 are related to how we think about resistance to either targeted therapies or immunotherapies. And ultimately, these 2 questions are related to how do we define what the optimal treatment for a patient with melanoma is. And that remains a very crucial question, and I'll show you some examples how we might think about going forward with that. The flip side to all of these treatments is that they have the chance of causing side effects. And I think it's important as we think about curing more and more people, how do we recognize who is going to develop side effects? How do we prevent these side effects? And then when these side effects do occur, how do we treat them in a way that's best for that patient? The 4th challenge I'm going to talk about is how do we treat rare, less responsive melanoma subtypes. So we know that certain types of melanoma are more responsive to targeted therapies and immunotherapies. And we'll talk about a few ways that might happen. And finally, I would argue that we do need to do a much better job in preventing melanoma in the first place. So let's zero in a little bit on each of these challenges. So the first challenge I'm going to address is how do we overcome resistance to targeted therapies? Well, one way would be to develop more potent inhibitors of existing targets. So we have 3 BRAF inhibitors. Can we improve on these BRAF inhibitors and extend their benefits? But I would argue that overcoming resistance is more likely to be achieved with using different combinations of drugs together. And I'll show you again an example of that. Another consideration is whether we need to be continuously treating our patients with these drugs. So right now, if you're being treated with a targeted therapy, the standard protocol is to continue this targeted therapy until it stops working. But there is some data to suggest that dosing, at least in preclinical models, that is more intermittent may have a benefit not only from the standpoint of reducing side effects, but also in extending the benefit of these treatments. And then finally, I think we need to broaden the mutations that we target. So right now, the only approved targeted therapies I mentioned target the BRAF signaling pathway, and that is only half of our patients with melanoma. So it's these sorts of research that I think will be useful in overcoming resistance to targeted therapy, and it may be even a combination of these approaches that ultimately prove the most successful. So I want to just show you one example from our own lab in which we find a combination of 2 drugs using a sequential dosing strategy can lead to pretty impressive results, at least in mice. So the combination involves a BRAF inhibitor called dabrafenib and a drug called S63845. These treatments don't matter so much as the concept I'm trying to bring. And if you can see that if we treat mice with melanoma, and we're treating them in this gray zone here, the S63845 really has no impact on the survival of these mice. In contrast, the BRAF inhibitor, shown in red, seems to control the melanoma while the mice are being treated with this drug, but shortly thereafter, the tumors continue to grow and unfortunately take the lives of these mice. But using a strategy where we combine the dabrafenib, the BRAF inhibitor, in the right sequence with this S63845, we can actually prolong the survival of the mice. And at least in mice, we can actually cure them. This obviously needs more work to reach the clinic, but I just show you this example as a possible strategy, and there are many, many others that I have not highlighted today. With respect to immunotherapy, we have numerous challenges. And one of the biggest challenges is that when you think about combinations, there are literally millions of combinations of drugs that you can think about. This is a study that looked at combinations involving immunotherapy between 2006 and 2019. For all cancer types, there were 3,000 clinical trials recruiting 500,000 patients. And so it will be very crucial to think about how what the optimal combinations will be for each patient, as it's likely that different combinations will be best for individual patients. And I think this highlights the need for our understanding of the underlying biology and biomarkers that help us figure out which of these patients should be treated with given therapy. So we know that certain combinations are effective. This is a treatment that is just recently published. It's a combination this is a combination immunotherapy with targeted therapy using these drugs atezolizumab, cobimetinib, and vemurafenib. And this combination of 3 drugs seems to show promising efficacy. We still don't know whether there's going to be long-term benefit from this treatment. And although this treatment has manageable side effects, the toxicity of this combination therapy is greater than each of these treatments alone. And so one question that remains to be answered is that whether the benefits of these treatments could be achieved by sequencing the therapies, or whether the treatments must be combined together to achieve the benefits that seem to exist. I wanted to highlight this other combination therapy. This is a combination therapy involving 2 drugs, ipilimumab and nivolumab. And I bring this example because this is now one of our best therapies of treating melanoma patients that have brain metastasis. This was considered very difficult. It has been a difficult problem in our field for a long while. And this treatment seems to have pretty comparable effects both outside the brain and inside the brain, and has now become the standard of care at our institution. So again, the first 2 challenges is how do we personalize the treatment? And ultimately, we need biomarkers to understand which treatments are going to be most effective in each patient. And this will include likely tumor-based biomarkers, such as the number of mutations that are present in your tumor, particular gene mutations, gene signatures, host-derived markers, such as the measure of your immune system, and even environmental influences, such as the bacteria that are in your gut. So how do we prevent, recognize, and treat side effects? Here we are severely lacking in understanding the underlying biology of these side effects. And just as I argued earlier with respect to improving treatment, I would argue that improving our management of side effects will come with understanding the underlying biology. So let me give you an example. This is a recent study where numerous patients developed a very severe type of skin rash. And what the authors of this study did was they biopsied the skin of these patients and determined, using a very sophisticated technique, the composition of each of the cells that were in the skin of these patients with this severe side effect. And based on this composition, they were able to identify a signaling pathway that was causing this rash. And in patients now who had this rash, using the identified pathway, using a drug targeting that pathway, the symptoms rapidly improved. So I think we need to be doing this not only for skin toxicities, but other toxicities related to immunotherapies and targeted therapies. Our fourth challenge that I'm highlighting is how do we treat rarer forms of melanoma? So one example are melanomas involve the eye, so ocular melanomas. This is a disease that has no approved treatments at the present time. 50% of patients who develop the melanoma in the eye develop metastatic disease. And the one-year survival from ocular melanoma is 15%. And that's a statistic that hasn't changed in 50 years. We know that ocular melanoma is distinct biologically from skin melanoma. They don't have the BRAF mutations, but they do have other mutations in genes such as GNAQ and GNA11, which is the subject of a lot of work preclinically as well as in clinical trials. For these patients, our current approach is to advocate a clinical trial. Finally, I think we need to do a much better job in treating or preventing melanoma. And we know that sun avoidance and barrier creams such as sunscreens, while essential, cannot be the only way to prevent melanoma. And that's because we know that the patients who are more likely, or the people more likely to develop melanoma, those with, for example, red hair or fair skin, can develop melanoma at least partially independently of ultraviolet light exposure. And so we really need to understand the mechanisms of melanoma genesis in these individuals and figure out ways of targeting these pathways. So as an example, we know that red-headed individuals have significantly higher risk of developing melanoma. This group developed a red-headed mouse model here. And they reasoned that if we can tan these red-headed mice, we could prevent them from developing melanoma. And you can see that this drug applied to the back of the skin of these red-headed mice, in fact, does tan these mice, although it remains to be seen whether this treatment can prevent the onset of melanoma in these mice. So I would argue that in order to design the next generation of therapies, we really need to understand the mechanisms of resistance. We need to understand side effects and the biological basis of these side effects. Ultimately, we need biomarkers that will guide us for selecting the therapy that's most likely to benefit specific patients. And as I mentioned at the beginning, it's research that will be the engine that will drive these advances, in my view. So I want to thank you all for your attention, acknowledging again my support, my lab, I don't know, am I supposed to take questions? I'm happy to take questions. Yeah.
You mentioned side effects, the rash.
Yeah.
Another side effect that we need to know about, besides the skin rash, what other side effects do we see?
Are you talking more generally or with particular treatment type?
The treatment that you were saying could create the rash. Is there other side effects that you put the patient on alert that could be happening?
Yeah, I think this is a great question. I think that each treatment has a risk of different side effects, and these side effects are different for individuals. For example, and they're different with respect to different treatments. So the side effects related to immunotherapy are going to be different than the side effects with targeted therapies. So I think when you're thinking about contemplating a treatment, it's very important to talk to your oncologist about what the specific side effects are for the treatment that you've selected. With respect to, for example, immunotherapies, immunotherapies activate your immune system, and when your immune system is revved up, really, honestly, any current part of your body can be affected with some side effects, be it the liver, the lung, your skin, your gut. These are all side effects. Some are more common than others, but I think it's important to recognize those side effects before you start your treatment.
Hey everybody, this is Cody really quickly. When you're asking a question, please do use the microphone because otherwise the folks in the live stream can't hear you.
Hi, this is a question from one of our online listeners. Julie's asking that with regard to rare melanoma, specifically mucosal melanoma, what new treatments are on the horizon? And what research is being done specifically for those with mucosal melanoma?
So I think that the question is related to mucosal melanoma, which I didn't talk about, but is one of the subtypes of melanoma that's less responsive to the treatments that we have. And that's partly for two reasons. One is that the mutations that are present in mucosal melanomas are different than we see with the standard cutaneous melanoma. So they don't have BRAF mutations. The other aspect about mucosal melanomas is that they seem to respond less well to immunotherapies than skin melanomas. And exactly why that is not entirely clear. But I would say that, I think that the advances in our treatment for mucosal melanoma will come down to understanding the genetic changes that are present in mucosal melanomas and why they also don't respond to the immunotherapy. So let me give you an example. So we know that mucosal melanomas don't have BRAF mutations, but they have mutations in a gene called c-KIT. And there are inhibitors of c-KIT. So these c-KIT inhibitors are currently in clinical trials. Several of them are in clinical trials for patients who have that particular mutation. So I think there's both progress with regard to targeted therapy options as well as combination immunotherapies that might be more effective for mucosal melanomas.
Doctor, for a melanoma patient who has been effectively treated with immunotherapy for an extended period of time, are there duration studies about how long they have to stay on that immunotherapy drug? What information do you have about that?
Yeah, so we don't have enough studies to answer that question. We know that it is safe in many patients to stop treatment. Exactly when will depend on the exact context. And in most patients who we have discontinued treatment because of a great response, most of those patients do not have a recurrence. But exactly the timing, which is, I believe, your question, is really unclear at this point.
Is this on? Hi. Could you comment a little bit on the intermittent dosing idea and specifically what that means? Are you talking about alternating treatment, so doing a BRAF/MEK inhibitor for a while until resistance and then doing something else and then coming back to it? Or are you talking about lower dosage or weekends off, or what's the thinking around
Yeah,
that?
great question. So the preclinical data would suggest that you can interrupt dosing and essentially take a holiday. And these studies were done in mice, so it's hard to correlate that to, a protocol for people. But generally, the concept would be you perhaps take you start your treatment, maybe takes 3 weeks of treatment, take a break for a week or so, and then resume. I can tell you that certainly I've seen patients who have been treated with targeted therapies and they develop a resistance. And I've had to take them off the targeted therapy, try other treatments. And if those treatments fail, I've gone back to targeted therapies and found that the targeted therapies do work. So this is all anecdotal. We need proper studies to sort of address exactly the timing and how we should be thinking about intermittent dosing. But it's certainly a very attractive idea, both from reducing the side effects of these treatments as well as enhancing their efficacy.
Hi.
Hi.
I'm Robin, with research advocate with OHSU and Soul Survivors. My wife was treated with IL-2 back in 2007. She ultimately passed away. But I remember how it affected her digestion. And you mentioned the gut bacteria, and I've seen some work on that. Can you proactively adjust someone's their gut bacteria to be prepared for the treatment to have a better result?
Yeah, so great question. Can you modulate the gut microbiome? So what we can say is that one can do this in mice, and in mice, modulating their gut microbiome can alter the efficacy of the immunotherapy. Where we don't know is whether that same approach can be used in people. And the challenge is that there are lots and thousands of gut microbiomes, and which one or two or 10 is crucial to modify your gut microbiome in a way that will enhance your therapy is really unclear at this point. There are a number of clinical trials testing this concept. We're using different combinations of microbiomes. I will say that as a cautionary note, because I get this question quite frequently from my patients, is there something I can do in terms of my diet to change the gut microbiome? And the answer right now is we don't know, but we do think there's been an association between probiotics and poorer response to immunotherapies. So this is something to be cautious about. I don't want to give the impression that you ought to go and try a probiotic to change your microbiome, because you may be doing harm and not good.
Hi, I'm Missy from Virginia.
Hi.
I wondered if you could talk about side effects that are that patients are experiencing more long-term, typically after they've had long exposure to immunotherapies or targeted therapies, about sort of, the good news is we're living longer, right?
Yeah.
The bad news is other crap is showing up.
Yeah.
Could you talk about those things a little bit?
Yeah. Yeah, so I think it's a great question. I think you've highlighted the issue of long-term side effects. And we know that many of the side effects that we see, for example, with immunotherapies or targeted therapies are short-lived and can be managed. But there are some rarer side effects that are much more difficult to treat. For example, we know that immunotherapies can affect the thyroid gland. And when you have in a lot of patients, we give combination immunotherapies, their thyroid gland no longer works, and they need lifelong treatment with thyroid replacement. That's probably not the end of the world, but there are certainly other side effects, neurological side effects that are extremely rare but do happen, and we've certainly seen our cohort of those at my institution, that are very hard to manage. and we don't have optimal treatments for them. And I think this gets to the question that we really don't understand why these particular patients might have side effects that are persistent and underlying the biology, because I think if we did have a better idea of those, we may have better treatments. But it's absolutely true that there are some long-term side effects, and these need more attention.
I have one here. Alex Harrington from Virginia.
Hi.
Piggybacking on Missy. So I am a patient who's underwent 5 protocols, and I see my side effects as I'm a Marine vet, so I see it as a battle wound. And I'm very fortunate to be alive, so thank everybody who's part of this. 6 years, still living. I'm just very grateful. So my question is, how do we help clinical physicians, either eye doctors or when it comes to our regular primary care, how do we help them help us? Because lately, we've been getting, especially with the latest side effects in the eyes, we've been getting the common responses, Alex, I just don't know. And this is regard to what's going on with the glands. My latest battle wound was the inflammation of lacrimal glands, where they had to do surgery through the eyelids. And yes, I look pretty tough with this eye patch. But how can we help them better help us? Because I know many physicians, they know their stuff. You guys go through, a ruling regimen when it comes to your training. But is there something that we can do to help them better help us as patients?
Yeah. Yeah, so this is a tough problem. So first of all, I'm glad that your melanoma appears to be treated. I think the side effects, first of all, we need to recognize that they exist and there are long-term side effects. I think that the way we can understand these side effects is by involving subspecialty care. So I think it has been common for melanoma oncologists to be involved in both managing your treatment and also your side effects. And I think what we need to move away, that's the paradigm we need to move away from. We need to move to a multidisciplinary team where we have rheumatologists, ophthalmologists, gastroenterologists who are dedicated to understanding these side effects. So, for example, at our institution, we have what we call an eye tox team. And that's eye, not like the eye, but eye tox. And this is a team that is composed of members from each of these subspecialties. So we have ophthalmologists, gastroenterologists, endocrinologists, rheumatologists. And I think by involving these subspecialty providers, we can learn more about what is the heart of these side effects. But also I think it helps you as a patient because you're seeing someone who's not seeing one of these side effects every year, but perhaps 5 or 10 a week. So I think that might be one paradigm, but ultimately I think in order to make progress, we need not only that, but we need to understand the biopsy. We need to understand the biology of these side effects, and that I would argue comes from our ability to sample your blood, your skin, or your liver, or wherever you might have side effects, because it's that biology that will help us advance our treatment for these side effects. So if there's one request I would make, if you were to have these side effects, do consider whether it's in your ability to allow us to biopsy your skin or your liver, or if it's safe to do so. And I think that would be one way we can make progress in this field.
We have five minutes left for questions, so if anyone else.
Hi, I'm Kimberly from South Carolina. I'm a patient. Where would we send those biopsies to?
Yeah. So I think that the first part of my answer to this gentleman's question was that I think we need specialized providers who see more and more of these patients with side effects.
Okay.
So I think that I think those are the providers where who have not only the clinical expertise with dealing with these side effects, but also have the academic research to understand what to do with these biopsies.
And also, what is some of the neurological side effects that you've seen with the targeted therapy?
With targeted therapies,
Mm-hmm.
We've seen generally the neurological side effects to targeted therapies are pretty minimal. We do see perhaps more neurological side effects with the immunotherapies, and those involve effects on any part of on your nerves. But is there something you're interested in with regard to
I've had the immunotherapy, and I've also had the targeted therapy. And
Yeah.
I've been on the targeted for 9 years now. And immunotherapy was 3 months prior.
Yeah.
I've also had 2 brain mets, and I am considered in remission. So we're on the fence about whether or not to continue the targeted therapy, long-term side effects, those kind of things.
Yeah, so we don't know of any specific neurological side effects with prolonged targeted therapy. I'm not aware of any.
Thank you, Doctor. Ian Schumann from New York. I'm also a patient. In your opinion, is it more the collection of the data? For example, we talk about all the permutations of different immunotherapy drugs. And how do you collect that data, both from people that don't react to it well and those who actually do get good results? Or is it the processing of that data? The artificial intelligence isn't there yet. How do you cleanse the data? How do you collect it? And what keeps you up at night? What is the biggest limitation in getting the data you guys need?
Great question. I think that it's probably both. So I think that, some of these trials, for example, with the combination therapies, even though the number of patients enrolled in these clinical trials is fairly large, a given institution might have, a small number of patients on that clinical trial. And so we do need the whole sum of data, but it needs to be analyzed, and often that data is in the repository of the pharmaceutical companies that run the clinical trials. And so I think that a tighter relationship between our academic research and our ability to get data from these clinical trials will be instrumental.
We have time for one more question, if anyone.
All right, and if you think of further questions, I'm sure Dr. Haq will be around, and you'll also be in the company of many other MRA-funded investigators throughout the day. But thank you so much, Dr. Haq. We really appreciate your discussion.